Good evening, everyone. We're going to take about another 4 or 5 minutes, allow people to connect.
Try to watch Batman, even if they're
Be another 2 or 3 minutes. Okay, we're going to start. We are recording. I'm going to share my screen. Okay. So, of the people who responded to the sampler exam, I scanned the essays and I chose one of the highest scoring essays and I circulated it. It is a part of the email invitation to this class. So you might want to check it out, see what that person scored. There's actually, I think, the later exams that came in, there were a couple of people that scored higher. Even the people who scored higher, check out the essay that I circulated. If you scored lower, you should be able to see why. If you scored higher, you should be able to see why. So you should be familiar with how we're going to proceed. So the way that we proceeded in the sample lecture and the sampler exam. So I'm going to give you the notes before time. We will put the notes on the screen at the class, and we'll go through the lecture, and then I'll provide you a highlighted version to show you the priority information. And the first time we're going to do this for real is tonight. Okay, on the screen, I have the schedule for the semester. I already got one question about this. If you take a look at this, you'll see what we're going to do and when we're going to do it. You'll see when the lab comes up. And on the 17th of this month, we will have a discussion about the scientific abstract. So you have to be familiar with the schedule. So I provided it once. No, actually, I did not provide it. I will provide it twice. So it's going to be a part of my next two class email, and it's already posted on the class's campus site. So if you go to the class's campus site, under announcements, you'll see all of the class emails, all of the invites. You'll also see the syllabus. You'll see the schedule and all of the PowerPoints to be used this semester are already posted to the campus site. You'll notice that prior to every exam, we have an open class pre-exam review. So you're expected to be familiar with the information in the syllabus and with the information on the schedule. So hopefully you're organizing the emails you received so they'll be retrievable, or you can depend on just going to the campus site and everything is there. So we can go into lecture mode. One second here. I might have some better notes here. Okay. I'm going to change this now. Okay, so a few words about this word, homeostasis. So you're going to see this word. Basically, the way it's used is for any process that keeps the body within physiological limits. I don't use it much, but when you see it, that's what it means. Basically, the reason I don't use it, when I first took biology, this word was used often, and sort of fell out of favor because the word implies a steady state. So if you just go to like the English dictionary, homeostasis is like a steady state, and that was never the case in the body. There are processes that can go up, that can go down, and still be in range. So a healthy person, you can have a temperature that's a little higher, a temperature that's a little lower. You can have blood pressure that's a little higher, blood pressure that's a little lower. And what's now referred to as homeostasis are the processes that keep vital functions within range, within narrow limits. So the reason that I'm using it here is because you're going to see it at other places. So this is sort of a word that sort of went out of fashion and sort of made a comeback. So it's like bell-bottom pants. It went out of style, now it's back. So this is going to be our first series of lectures, and this is going to be more than half the semester. Blood, heart, circulatory system, lymphatic system, the immune system. So we're going to talk about blood. And to show you, these PowerPoints are all loaded. And I believe that during the course of this lecture, I referred to about 3 of these slides. But the source of these slides are per chapter. So this is not your recommended textbook. This is a textbook from a different school. But these PowerPoints became available, so I'm using them. But they're per chapter. So I'm going to use about 3 or maybe 4. And they're like 46 slides here. So you may want to be aware of which slides that are being referred to. So if you need to pick them up by going to the Canvas site, you'll know the ones that were emphasized. So blood is the liquid of life. So you have a fluid flowing through tubes. The tubes in the circulatory system, the life-giving fluid is blood. And at some point in the circulatory system, they're connected to a pump. The heart is forcing this fluid to go through these tubes that transit the entire body. Blood has living elements and non-living elements. The living elements are solid, the non-living elements are liquid. The living portions of blood, so these things are alive. Blood cells and cell fragments. Red blood cells, white blood cells, platelets are fragments. Okay. And I'm going to say this about 12 times between this lecture and next lecture. Platelets are not cells. They're pieces of cells. They're cell fragments. The cells are white blood cells and red blood cells. These living portions are swimming in liquid that's not living. So the non-living liquid portion is plasma. The living elements are the blood cells and the cell fragments. For the second time, platelets are fragments of cells, not cells. As we go through this, and this is one of the reasons we did that sample exam, you'll get used to my shorthand. It's a specialized connective tissue. So this is something everybody should know. The specialized connective tissue. I'm going to do this. I want to put this aside for just 1 second, and I want to briefly go over the lecture we had. Well, actually, I'm not going to do that. I'm going to continue. Okay, so this is connective tissue, a specialized connective tissue. It's called that because the stem cells that can develop into connective tissue also can develop into blood cells. But blood is a lot different than connective tissue, so these are called one of the specialized connective tissues. Terms you have to know. Red blood cells, white blood cells, platelets. Erythrocytes, red blood cells. Leukocytes, white blood cells. Thrombocytes are platelets. And I think I'm up to the 4th time already. Platelets are not cells, they're cell fragments. Okay. Also, you see, how I do this, PowerPoint chapter 19. Keep in mind, these are the PowerPoint chapters, not the chapters in the recommended text. So here, referring to PowerPoint page 4, this is PowerPoint chapter 19, and this is slide 4. Red blood cells are erythrocytes. White blood cells, leukocytes. Platelets, thrombocytes. Erythrocytes, leukocytes, thrombocytes. Red blood cells, white blood cells, platelets. So we're going to talk mainly about erythrocytes. Erythrocytes make up about 40 to 45% of your blood. So you take all the stuff in your blood, about 40 to 45% of them are your red blood cells, erythrocytes. And that is shown here. Voila. Okay, so this picture shows down the breakdown of blood. And there's the erythrocytes. They're going to make up about 45% of the blood. That percentage is called your hematocrit. So when you go to a medical facility they check your red blood cell count, it's going to be, unless you're really sick as a dog, it's going to be somewhere between 45% and 40%. And that is remarkably stable, meaning if your hematocrit is 41.5% now, 3 months from now, it's going to be 41.5%. 9 months from now, it's going to be 41.5%. So, because leukocytes, thrombocytes are only actually like 1%, this is the vast majority of your blood cells. Your solid elements are the erythrocytes. And the rest of it is the plasma, the non-living portion. So these are some characteristics. It's much thicker than water. So this also, you can see my shorthand. 5x viscous than water. So it's 5 times thicker than water, about 8% of your body. The average person, guy's not too big and not too small, is going to have about 5 liters. Because the blood travels in the circulatory system, it travels in a series of tubes. There's a pump pushing it through, contract, relax, never stopping. The blood is in constant motion. And one of the results of it being in constant motion is the elements of the blood are really evenly distributed. I have the major function of the blood up front. Transports respiratory gases. That's oxygen, carbon dioxide. Oxygen, you need oxygen. All your tissues need oxygen. Okay. So for them to make energy, they need oxygen to burn the nutrients. So you eat, you intake nutrients. They have to be processed. Those nutrients become energy to keep you alive, keep your tissues alive. To burn those nutrients, you need oxygen, respiratory gas. It's picked up in the lungs, dropped off at the tissues. The waste product from this process is carbon dioxide. Also travels in the blood. So it transports carbon dioxide and oxygen. Transports the nutrients, the metabolic waste, the white blood cells, and we're going to have like plenty of information on that. That's the immune system. You should know what pH is. pH of blood is in narrow limits. Healthy person, the pH of blood, always within narrow limits. Distributes heat. Depending on if you're like a small person or a big person, it takes an average of about 40 minutes for blood to circulate. So blood will leave the heart, go into arteries, capillaries. So it goes from the heart to the lungs to the heart. In the lungs, it picks up oxygen. Oxygen-rich blood leaves the heart, travels in arteries and capillaries, then veins. Started out as oxygen-rich. By the time it gets to veins, it's now oxygen poor. And it's going to make its way back to the heart. That trip takes about 40 minutes. So, items traveling in the circulatory system will reach their target within about 40 minutes. So you should have had endocrinology in AMP 1. And you should know something about hormones. So hormones come in small quantities. They're really specific for their target. And it's going to take about 40 minutes or less for a hormone to reach its target, because that's the amount of time it takes blood to circulate the body. We're going to see this like next lecture. Blood is also involved in coagulation. So, the living portions of blood: erythrocytes, leukocytes, thrombocytes. Thrombocytes are platelets. Platelets are not cells, they're cell fragments. The non-living portion of blood. Okay, I have a note here, PowerPoint page 7. I think I'm not going to use that. That's just a list of items in the blood. I'll show it to you. So this is just a listing. This is just a listing of all the things that are non-living in blood. So it's about 45% of blood are the things that are living. The non-living is about 55%. So you have the living elements floating around and basically a water environment. Plasma. So if you receive plasma, that means you receive a transfusion of blood with all the living elements taken out. They take out the erythrocytes, they take out the leukocytes, they take out the thrombocytes. The plasma is all of the liquid and the things floating in the liquid. So the things floating in the liquid, they're dissolved. They're dissolved by the liquid. So the liquid is mostly water. That's a solvent. So it dissolves these items. These items are dissolvable. The things that get dissolved are the solutes. The water portion of plasma is a solvent. The things dissolved in the plasma are the solute. And that's like universal. So if you're in a nail salon and they're using acetone on your nails, then the acetone is the solvent. The nail polish that's being dissolved is the solute. There's about 100 different items that can be dissolved in blood. The nutrients, proteins, carbs, fats. 1 point for carbohydrates. I use this formula, and mostly when I use it, I refer to glucose, which is the simplest, which is among the simplest carbohydrates. If there are any chemists in this class, they know that it could be glucose, it could be some other things. So all the simple carbs have this formula. When I use it, I'm going to be using it for glucose. So we're describing the things dissolved in blood. You can have nutrients. You can have the gases we mentioned, oxygen, carbon dioxide. You can have these charged particles. You should be able to recognize the notations as chemicals: sodium, magnesium, potassium, chloride. This one you're going to see bicarbonate, phosphate. So these are all ions. They're not balanced. The positives and negatives are not balanced. So again, we're talking about things that could be dissolved in blood. The proteins, plasma proteins, proteins dissolved in blood. These are notable. Of all the items dissolved in blood, of all the solutes, the proteins will make up about 7 to 8% of them. But the proteins have a big impact. They have an outsized impact because of their size. Proteins are too large to move across tissues. So they can't get across the cell membrane. They can't get outside the circulatory system. They can't get inside the circulatory system. They're too large. They're too large to move. So even though they only make up 7 to 8% of the dissolved items, they have a big impact because they themselves are too large to move. Most proteins are large, too large to move. There's medium-sized proteins, they're too large to move. The small proteins are too large to move. They're not many teeny tinies. So among the proteins, there's hormones. This is the most common, albumins. Most of the plasma proteins are albumins. Globulins, this one we're going to come back to. This is antibody, gamma. This one you're going to see next lecture, fibrinogen. Fibrinogens and clients. So these are different types of proteins that can be dissolved in blood. Some proteins act as carriers, meaning you have items that need to get from point A to point B. They're going to travel in the blood. The problem is they can't dissolve. They can't become solutes, except when they are tied to a carrier protein. There's some proteins whose job it is to pick up other items, and together they're soluble. Once it dissolves, it can float around and go from point A to point B. Okay. So if you get used to my shorthand, cell membranes impenetrable to almost all proteins. Proteins in blood confined to blood. So the proteins inside can't get out. Proteins outside can't get in. The proteins have an outside impact on what moves in and out, but they themselves can't move because they're too large. And we'll get more of this later. We'll talk about osmosis. We'll talk about osmosis is the movement in and out of the cell membrane. And what we're talking about is in and out of the circulatory system. What's moving is the liquid, is osmosis. It's what's moving is the solute. If things dissolve, that's diffusion. And the driving force of that movement is called hydrostatic pressure. We're going to come back to that. If it's the liquid, if it's the dissolved items, it's called colloid osmotic pressure. We'll come back to that. Okay. We're going to get to the good stuff. Okay, you'll notice blood cells. All blood cells, the site of production is the red bone marrow. The red bone marrow produces all blood cells. That's lymphocytes, leukocytes, thrombocytes that are not cells but cell fragments. I think I said that like 6 times already. The process of making any blood cell, hemopoiesis. So here you see there's an alternate term. But either I'm going to go with the term that's more popular, or I'm going to go with the term that I learned back in the day, either or. So making blood cells, the general name is hemopoiesis, blood cell production. It occurs in the red bone marrow. All blood cells up in the red bone marrow, and they start off as a stem cell. Stem cells that can become any blood cell, hemocytoblasts. And you'll see we have some different terminology here. Okay, so this is a stem cell. They call it something different. But this is the stem cell. It's in the red bone marrow. It can become any blood cell. So it becomes a erythrocyte is going down this way. It's going to become a platelet. A thrombocyte is going to go this way. It's going to go to one of these white blood cells. But they start off as a stem cell. They can become any cell. So you need to know the term hemocytoblasts. So this just describes the bone marrow. When you're young, it's all your bones can do it. When you're older, your long bones can do it. That's the amount of red blood cells produced every single day. The general term, hemopoiesis, the stem cell hemocytoblasts, replaces the red bone marrow. If you're talking about red blood cells, erythrocyte, hemopoiesis is blood cells, generally production of blood cells, production of erythrocytes, erythropoiesis. So, if you start off the stem cell, anything going down is hemopoiesis. But this one is erythropoiesis. This is going to be a red blood cell, not these other things. Hemopoiesis is a general term for all this production from the stem cell, hemocytoblasts. But if you go this way, that's erythropoiesis. It's going to become a red blood cell. Okay. Here. Okay. So the item that forces erythropoiesis, that forces the hemocytoblast down a particular path, down the path to become a red blood cell, to make it erythropoiesis, is a hormone. EPO. Here it is. So it's hormones from the kidney or the liver. So your blood cells are low. The EPO kicks in. EPO forces the hemocytoblasts to go down this way to become an erythrocyte. So this is a controlled process. This is a regulated process. You need more red blood cells. Your body detects it. It produces more hormone. The hormone forces the stem cell this way. Erythropoiesis. Once again, make sure we know what we're talking about. What is that? There's a stem cell. Hemopoiesis is going to become something. Development from the stem cell is hemopoiesis. Stem cell, I'm calling it hemocytoblasts. But to get it to come this way, you need a hormone, EPO. EPO makes it do erythropoiesis and become an erythrocyte. And you see it here. Stem cell, it becomes committed. So when it's a stem cell, it can become anything. Now it's forced down a particular way. It becomes committed. That's just like getting engaged. You couldn't marry a lot of folk, but then you get engaged. That forces you down here, and then you're going to get married, and then you're done. So, EPO will force the stem cell down the path to become an erythrocyte. When it becomes committed, when it can only become like a pre-erythrocyte, it develops items on the cell membrane. So cells always have like a bunch of stuff sticking off the cell membrane. It's like the roof of your house has something sticking off of it. It has a chimney. It has an antenna. It has the doohickey for your cable, satellite dish for your cable. So your roof has things that stick off of it. Cell membranes have things that stick off of it. Extensions. When the stem cell becomes committed, it now has the items sticking off the cell membrane that are particular to erythrocytes. It becomes committed. It's like a baby erythrocyte. It develops and develops and develops. The immature stage, another word you need to know, reticulocyte. The stage before a fully developed erythrocyte is a reticulocyte. Stem cell becomes committed. It's young. It's a teenager. It's an adult. You need to know reticulocyte is the immature stage right before full maturity. It's a erythrocyte. One note here. EPO. EPO forces your body to make red blood cells. It forces those stem cells down the erythropoiesis path, and you make red blood cells. This is what Lance Armstrong was doing. So Lance Armstrong was the biker who was winning year after year after year after year. The Grand Prix, whatever the event is called, in Europe, I think it's in France. It's impossible to win year after year after year, but he was winning year after year after year. He got busted. He was shooting himself up the butt with EPO. So when everybody else gets tired out, he has more and more red blood cells. Red blood cells carry oxygen. So he got caught, he lied, and he got trapped. It's telling the truth. Always happens. Page 11. Okay. At the reticulocyte stage, the immature red blood cell stage, the immature erythrocyte stage, the reticulocyte starts producing an item called hemoglobin, synthesized. Before and during reticulocyte stage, it synthesizes hemoglobin. I'm going to show you this. Make sure you know how to spell it. Here it is, hemoglobin. So even before it's fully mature, it's in reticulocyte, it starts to produce this hemoglobin. It is pumping out hemoglobin. That's what it does. Okay, a bread factory makes bread. They pump it. This is making hemoglobin. That's what reticulocytes do. That's what reticulocytes do. They make it and make it and make it and make it. So they're very, very active. All their inners, the cell organelles, are working hard, and they're pumping out this hemoglobin. When the amount of hemoglobin within the reticulocyte gets to the point where it occupies about 34% of the interior of the reticulocyte. So the hemoglobin is not going anywhere. It's not leaving the cell. Okay, so the bread factory, there's no trucks. You got more and more bread. Okay, it's just like a hoarder. You know, you're saving stuff and saving stuff and saving stuff. Your apartment's not getting any bigger. The reticulocyte's not getting any bigger. In fact, the reticulocyte's getting smaller. It's filling and filling and filling with hemoglobin. At the point where hemoglobin is about 34% of the weight of the cell, two things happen. This is the point where the reticulocyte leaves the red bone marrow, enters the circulatory system. So when it enters the circulatory system, it's still a young reticulocyte. It's not mature yet. The other thing that happens is from this point on, more and more and more hemoglobin, you start to choke off its inners. It's like a hoarder, okay? You save stuff and save stuff and save stuff and save stuff, and then to get more room, you get rid of your refrigerator. And then to get more room, you get rid of your kitchen table. And then this is what happens. It's hoarding hemoglobin. The nucleus starts to shrink. The overall cell starts to shrink. The inners of it start to shrink and be ejected. So when there's enough hemoglobin to make up about 34% of the cell, it leaves the red bone marrow, enters the circulatory system, and it starts to impinge upon its inners. There's so much hemoglobin now the nucleus can't function. The inners can't function. Ribosomes, these things can't function. They're just getting crowded out. Okay, so reticulocyte migrates through the walls, enters the capillaries. The reticulocyte is in the circulatory system. It's going to be another 3 to 5 days before it fully matures. It leaves as a teenager. It fully matures while it's in the circulatory system. When it's fully mature, it has no more inners. Okay? It's the hoarder. No more organelles. The nucleus has been shrunken and expelled. Organelles shrunken and expelled. Its inners, cytoplasm, it's full of hemoglobin. There's so much also the size of the red blood cell of the erythrocyte has shrunken. Okay, it has no more inners. It sort of collapses in on itself. It's like a poker chip, where the middle on both sides is indented. Just a sack of hemoglobin. About 1/4 of a billion hemoglobin molecules in one erythrocyte. Essentially, it's a sack of hemoglobin. So it is now super small. I don't know where do I have this thing at. Anyway, the mature erythrocyte is super small, the smallest cell in your body. Like somewhere in this notes, I have 5 to 8 microns. Very, 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 very small. Very, very, very small. The middle is collapsed on both sides like a poker chip. Packed with hemoglobin. 1/4 of a billion hemoglobin molecules in each erythrocyte. So this is what's going to be carrying the oxygen. This has to swim in the blood. It has to pass through tight terms. It has to get past the capillary. Capillaries are really, really small. So if the red blood cell is about 6 to 8 microns, the capillary is smaller than that. But there's a certain protein that's a part of its cell membrane that makes it super flexible. It can squeeze through spaces. So if the erythrocyte is about 6 to 8 microns, if the diameter of a small capillary is like 5 microns, it can squeeze through because it's super small, super flexible. It's like that joker. When I first got married, we left the bathroom window open, just a crack. Came back home, no television. Somebody just slipped their way right in. So when it's young, a young erythrocyte is highly active. But by the time it matures, has no more inners, it is super small, it's biconcave, like a poker chip. The middle on both sides has collapsed. It can't produce much energy. So if you remember this from general biology, metabolism, 3 stages: glycolysis, Krebs cycle, citric acid cycle, electron. That's how you make energy. Well, it can't do that. It doesn't have any inners. It can only do the first step of metabolism because that step does not require oxygen. So if the erythrocyte is anaerobic, doesn't burn oxygen, that's a good thing because the job is to carry hemoglobin. The hemoglobin is going to carry oxygen. So the erythrocyte is not going to consume the oxygen that it carries. It doesn't have mechanisms to do that. It's only doing the minimal respiration, which is glycolysis. Okay. red blood cell structure is highly adapted to its function. Super small, it can type capillaries. The hemoglobin is going to pick up oxygen, give up oxygen, pick up carbon dioxide, give up carbon dioxide. All of its inners are close to the surface. So things can get in and out. It's anaerobic. Okay. Now the complicated portion. So this is the erythrocyte, super small, smallest cell in the body. This is fully mature. The center on both sides has collapsed. No more inners, packed full of hemoglobin. 250 million hemoglobin molecules. So this is a hemoglobin molecule. So to explain this, let me just okay. Hemoglobin, two portions, the globin and the heme. The globin is a protein, the heme is a pigment. Hemoglobin, the globin plus the heme, the protein portion. So protein are made up of amino acid, amino acid, amino acid, amino acid, amino acid, amino acid, chain of amino acids. What a protein can do depends on their three-dimensional shape. So this protein, globin, is made up of two chains, amino acid, amino acid, amino acid. I'm sorry, four chains. Four chains of amino acid, 2 and 2. So that's what this tries to show. 2 light blues, 2 dark blues. 4 chains. Each chain has 1 heme. The yellow disc are the heme. Each heme has 1 iron. Fe is iron. Okay. 4 chains. Each chain has 1 heme. Each chain has 1 iron. Iron is Fe. Let's blow this up. You have 2 light blues, 2 dark blues, all squished up, 4 chains. Each chain has 1 heme, the yellow disc. Each heme has 1 red spot, the iron. The iron is the oxygen binding site. So 1 molecule of hemoglobin can bind 4 oxygens. The red disc, the iron is the oxygen binding site. Complicated protein. If you can see this formula, the formula of hemoglobin. Notice at the end, iron, 4 of them. 4 chains, each chain has a heme. Each heme has a globin. Each globin has 1 iron, 4 irons per molecule. So this hemoglobin, these 4 chains, you have, every chain is amino acid, amino acid, amino acid, amino acid, amino acid, amino acid. every amino acid has to be in the proper place. So if you take one amino acid, it has to be a certain distance from the one above it, a certain distance from the one below it, a certain distance from the one to the left, a certain distance from the one to the right. Every single amino acid has to be in the exact place for it to work. This is like spaghetti on a plate. And every piece of spaghetti absolutely has to be in the right place. So it's the iron of the heme that binds the oxygen. Oxygen is carried in the blood on the erythrocyte. Every erythrocyte has 250 million molecules of hemoglobin. Every hemoglobin, every one of the 250 million has 4 hemes. Every heme has a globin. Every globin has 1 molecule of iron. Every molecule of hemoglobin can carry 4 oxygens. And there's 250 million hemoglobins in every erythrocyte. 4 times 250 million is 1 billion. Every erythrocyte can carry about 1 billion molecules of oxygen. Okay, so some of the cleanups here. So you're dependent on the iron binding sites, but iron is toxic. So any iron in your body has to be bound to a protein to make it soluble. If it's not, it's going to poison you. So you're going to have the iron down to hemoglobin, or it's going to be stored. There's another protein that stores it. And to get it from place A to B, there's a different protein that transfers it. But the iron cannot be free. As we bound to hemoglobin, or bound to a storage protein, or bound to a transport protein. It's the iron that picks up oxygen and lets off oxygen. So it picks up oxygen in the lungs and lets off oxygen at the tissues. This is how your tissues stay alive. Because your tissues are close to the circulatory system. The circulatory system contains blood. The blood contains hemoglobin. The blood contains hemoglobin. The hemoglobins contain heme with an oxygen binding site that can pick up the oxygen and then drop it off. So it picks up the oxygen at your lungs. It becomes oxygen-rich blood. It can drop off the oxygen at your tissues, oxygen-poor blood. The affinity, the attraction between oxygen and the oxygen binding site in the hemoglobin is super high. Okay, if they're around together, they're going to get together. Okay, you can't have boy and girl, or some hanky-panky's going to go on. The affinity, the attraction between oxygen and the heme binding site is so high that when your oxygen poor blood travels to the lungs to become oxygen rich, doggone near every single possible oxygen binding site is taken up. At the lungs, 90% of every single oxygen binding site in every single red blood cell is taken up by oxygen. The attraction is so high that even oxygen-poor blood is going to have 75 to 80% oxygen. Oxygen-rich blood is almost 100%. Oxygen-poor blood, you still got a lot of oxygen. So the next 2 points I'm going to give you. How does oxygen transport in the blood? And how does carbon dioxide travel in the blood? You could get these now, and you're going to get them now, or you could get them when we do the respiratory system. But why wait for the rush? We'll do it now. So how does oxygen travel in the blood? This one is straightforward. Almost all of it is in hemoglobin. A little bit, 1.5%, can dissolve. A little bit of it dissolves as a solute, 1.5%. Almost all of it is carried in hemoglobin. That's how oxygen gets around. And this just shows that it's picked up and dropped off. Picked up at the lungs, loaded at the lungs, dropped off at the tissues. Oxygen loading occurs in the lungs, oxygen unloading occurs in the tissues. This just has to do with those 4 polypeptide chains that make up hemoglobin, the spaghetti on a plate. Every single portion of each of the 4 strands has to be in an exact place. If one oxygen binds, then the chances of the other 3 binding sites getting oxygen goes up. If 2 oxygens binds, and the chances of the other 2 binding sites getting oxygen goes up. Every time you add an oxygen or take away, you're changing the shape. So you're changing the characteristics of it a little bit. This is a problem. Carbon monoxide and cyanide. So the attraction of oxygen for the binding site in hemoglobin is super high. But carbon monoxide is even higher. Carbon monoxide is going to outcompete oxygen. Cyanide is going to outcompete oxygen. So my poor analogy here, you can have high attraction. Okay, so you can have a couple, they're madly in love, they're super attracted, they're engaged, they're fiancés, they go out to dinner, and who is at the next table making eyes at your boyfriend? But Beyonce. That's the problem. You got even higher attraction. So if you're like in the garage and you're sniffing the tailpipe, that's going to outcompete oxygen for those binding sites. If you're taking cyanide pills because you're a Nazi and you've been caught in World War II and you want to kill yourself, that cyanide is going to outcompete the oxygen. The bottom line here, how does oxygen travel in the blood? 98.5% of it is on the hemoglobin, and a little bit is going to be dissolved. How does carbon dioxide travel in the blood? 3 ways. 7 to 10% will dissolve. 20 to 30% will bind to hemoglobin, but on the globin, not the heme. It's not going to bind to the yellow disc of the heme or the red dot of the iron. It's going to bind somewhere on the blue. So how does carbon dioxide get around? I mean, 10% will dissolve, 30% will bind to hemoglobin. On the globin, not the heme, so it's not going to compete with oxygen. The rest of it will be converted to an item that is readily dissolvable, bicarbonate. So you got this little formula here. As soon as there's carbon dioxide, it's going to be converted to bicarbonate. The name of the intermediate, the name of the enzyme that does it. So one word about enzymes, you should know this already in AMP2. Almost everything chemically that happens in your body happens because enzyme pushed the reaction. This is hard to do in a laboratory. It happens all the time in your body. You get some carbon dioxide dissolve, some carbon dioxide and hemoglobin, and the rest of it is readily converted to bicarb. Hard to do in a lab, but easy to do in your body because in your body you have an enzyme that makes the formula go. So to details, the carbon dioxide that travels this way, that gets converted to bicarb. There's a mechanism that stuffs the erythrocyte full of bicarb. It's just not just high concentration, the low, it stuffs it in. It's not just diffusion. There's a mechanism that stuffs this stuff in. Okay. So chloride shift just means bicarb is packed, despite a concentration gradient, is packed into erythrocytes. This Haldane effect. Haldane effect has to do with these. The carbon dioxide that travels on hemoglobin, on the globin, not the heme. Haldane effect just says you have oxygen on the heme at the ion binding site. That's carbon dioxide on the globin. They're at different places, but they have a certain relationship to each other. This goes back to the spaghetti on a plate. You move any portion of one strand and you change the molecule a little bit. You add an oxygen, you take away an oxygen. You add a carbon dioxide on the globin, you take away. This is going to change the properties. So the more carbon dioxide, the more, let's say it this way, the more oxygens, so you can have 1 or 2 or 3 or 4 oxygens. The more oxygens, the less carbon dioxide can bind. The less oxygens, the more carbon dioxide combined. It has to do with the shape of the molecule. The point is, although the oxygen is at one place, the carbon dioxide is at the other place, they have some relationship. The more oxygens, the less carbon dioxide. The less oxygens, the more carbon dioxide. Okay, so we're getting to the end of this story here. So we started off with hemocytoblast. EPO was high, making the stem cell go through erythropoiesis, becoming an erythrocyte. Goes to the early stages, goes to the reticulocyte stage. The reticulocyte stage, it starts producing hemoglobin. It leaves the red bone marrow. It's in the blood. It pushes out its innards. After 3 to 5 days, the reticulocyte becomes an erythrocyte. Really, really small, packed with hemoglobin, no innards. How long can it last like that? Squeezing through really small spaces. Erythrocyte life cycle, 100 to 120 days. Can't last forever like that. You have no innards. You can't, you can't really take nutrients and make it into energy, not very well. Can't live forever. The erythrocytes will live for 100 to 120 days. They have no innards. They can't do anything. They can't repair themselves. This is called when cells die when they're supposed to die, apoptosis. As opposed to your cells dying because you got COVID, the cells dying because they got murdered, the cells dying because you had an accident and fell off the Ben Franklin Bridge. Okay. If they die when they're supposed to die, it's called apoptosis. So an erythrocyte will live, when it's a full erythrocyte, it's going to live for about 100 to 120 days, and then it's done. So when it dies, it gets eaten up by the immune cell. But some of its material gets recycled. A lot of its hemoglobin gets recycled, goes to the liver. The heme becomes the pigment that is responsible for the color of stool. The iron is mostly recycled. Keep in mind, when the erythrocyte breaks down, the iron can't be free. Free iron is toxic. It has to be bound to a protein to transport it, or it has to be bound to a protein that stores it. So, last point of the lecture. So you make red blood cells, and then the red blood cells die. Making the red blood cells, that is a control process. EPO, there's a hormone. When you need more, you get more hormone. That hormone forces the stem cell to become an erythrocyte. So the production is controlled. The death rate is not controlled. They die when they die. They run out of steam and they die. But the balance between their production and their destruction is so precise that your hematocrit doesn't change. So you have a hematocrit telling you have 41.5% red blood cells, If you're healthy, 3 months later, you can have a hematocrit of 41.5. A whole bunch of blood cells have been born. A whole bunch of blood cells have died. Your hematocrit has not changed. The production is controlled. The death, they just die when they die. So we have come to the end of this first lecture. And our method will be, I will give you these notes ahead of time in the invitations to the Zoom meeting. We will review the notes in class. In the invitation to the following Zoom meeting, I will give you the highlighted versions. So the purpose of the highlighted version is for you to see the lecture and see what my priority information is. Some of the terms that we use, you won't hear them out of me again. Some of the things we talked about, you're going to hear them over and over, and you're going to see them highlighted. That gives you an idea of where to prioritize your studying and gives you an idea of how the exams are going to be built. So we have like 14 people connected. We have like 23 people in the class. So hopefully the balance will pick all this up on the YouTubes. So just one quick comment. Unlike the two other schools that I teach at, Camden County College does not have a contract with Zoom. So we are using the publicly available Zoom. So it's going to be more a pain in the butt than it usually is. In addition, I cannot record to the cloud. Okay, so the other schools, at the end of the meeting, I get an email, and the email has a link. You can follow that link, and you can access the recording. Or you can just go to the Zoom site and you have all the recordings. Well, we can't do that. So what I do is I convert, I save to my computer and convert it to a YouTube. So what you'll have is a link to the YouTube instead of a link to the Zoom recording. So the people who are not able to connect will have to depend on the YouTube recording. It takes a little bit more effort for me, but that's all we got to deal with. Okay, so I guess we're about done. One more thing. During the entire course of the semester, if you need me to repeat something, if you need me to spell something out, if you see some shorthand on my cards you don't understand, interrupt me and ask me. I had one student at a different school last semester who claimed to be a super introvert. She always had questions that came right after the lecture in an email. She never opened her mouth during class. So you can write me an email, or even better, ask a question in class so everybody can hear it, because they probably have the same question. Okay, so that's it for the first class. Make sure you pick the schedule. I'm going to distribute it in the next class, the next two class emails, and it's already posted on the campus site. Okay, we're done for tonight. I don't know if the snow is still coming down, but I'll try to stay warm.
Professor Hayward, is there a moment for questions or?
Right now, do it.
Okay. Regarding the schedule, I'm So I'm used to, I'm also a student at CCP this semester, but the way it works with the schedule, you have a class and then maybe 24 hours or whatever it might be, 3 days later, something's due. So if we have the lab, let's say in 2 weeks, is it due 24 hours or like from when it's posted? Or can you elaborate on that a little bit?
Okay, so the assignments we'll have. Let me. I'm going to share the screen again to put up the schedule. Okay. So the labs. The accountability for the labs will be a lab report. We are going to have no face-to-face labs. We're going to have a series of videos. So you look at some other guy doing the lab. You watch the video, and the accountability will be a lab report. Super simple. When we have the first lab, I'm going to spend time on the format of the lab report. It's going to be super easy. The lab reports are due. You can turn them in at any time from when the lab occurs. So the first lab is on the 15th.
Yes.
You can hand it in on the 15th, or you can hand it in the next class, or you can hand it in the next week. As long as I see all lab reports by the next to the last day of the semester. So you can give it to me as they go, or you can give them to me all at the last minute if you're a last minute kind of person. And keep in mind, the labs are 10% of your grade. You do the labs, you get 100. You don't do the labs, you can't pass the class. So I need to see 5 lab reports by the next to the last day of the class.
Okay.
So we got lab number 1. After lab number, these classes, after lab number 1, they're going to do like 15-minute classes. It's just a video. So after you see the format, it's going to be real simple. And then you got lab number 2, and you got lab number 3, and lab number 4, and lab number 5. Once there's a lab, you can hand in the report. Email me the report at any point, as long as I have all 5 by next to the last day of the semester. If I get a lab report that's jacked up and has been really, 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 really bad, then I'll just ask you to fix it. And then you'll have your 5 labs, you get 100. But you absolutely have to do all 5 labs. And I had an email question already about our other assignment, the abstracts. So on this day, I'll give you a whole bunch of detail on the abstract. So all it is reading 3 articles. You read the article and you write a really, really, really brief summary. So usually in a scientific article, before the article starts, there's a little summary of the article. So if you're like in some sort of scholarly magazine and you have this long-ass article and you want to figure out, should I read this or not, you read the little summary, the abstract. That's what an abstract is. It's a 1-paragraph summary of a much longer article. So I'll give you the source. You'll have the archive. There's hundreds of articles. Any 3 articles would do. Articles on the same thing, articles on different thing, articles on sports medicine, articles on dinosaurs, articles on foot fungus. It doesn't matter what the article is. But between this date and the next to the last day of the semester, I need to see your scientific abstracts. On this date, I'll give you more details. I'll give you like the rules. You're gonna need like an editor, but I'll give you the resources. It's not going to be hard to do. But you do it, and I'm just going to check the syllabus to make sure I'm telling you the right thing. So, where are you? Here you go. You do it, and you get 15%. So again, I'm not going to, the abstracts, I don't really grade them, except done or not done. If you give me something that's super jacked up, you just fix it up. So you do the abstracts, you get it in on time, and you get 100. And that 100 is time 1.150. 100 times 1.15 is 15 points. So you get 10 easy points for the lab, you get 15 easy points for your abstract. That's going to help your grade. And the answer to the question asks, when are they due? They're assigned on this date, and they're due by this date. I used to have things due at the last day of the semester. Didn't work because inevitably somebody's going to give me something that's really, really jacked up. And when it's the last day, it's the last day. So I end up accepting some really messy stuff. So anything that comes in here and it's a total mess, I can always say, give it to me next class. Fix it up and give it to me next class.
Awesome.
Okay,
Thank you.
so this, okay. So the schedule, I'm going to distribute it in the next 2 class emails, and it's already posted in the classes campus site. Okay, I think that's it. Thank you.
Thank you. Have a good evening, everyone.
You.
Thank you.
Thank you.